Hi, I'm Elizabeth Bowen, conference producer at Technology Networks, and I'm pleased to introduce our next talk towards the making of molecular movies directed by the Circulating Proteome. Please join me in welcoming Professor Jochen Schwenk. Jochen is a professor in translational proteomics at KTH Royal Institute of Technology in Stockholm, Sweden, and holds a PhD in biochemistry from the University of Tübingen in Germany. Jochen is an elected member of HUPO's Council, chair of HUPO's Plasma Proteome Project, scientific director of SciLife Lab's Affinity Proteomics Unit, and part of the Human Protein Atlas Management Group. His expertise extends to pioneering novel aspects of the biomarker analysis pipeline, encompassing diverse domains like study design, pre-analytical variables, microsampling techniques, assay development, data integration, and the exploration of emerging translational avenues. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. I will now hand over to Professor Schwenk to begin his presentation.
Thank you very much. It's a great pleasure to present to you some novel aspects of the research that I'm conducting. And while thinking about this presentation, I sort to switch from the normal scientific opening to maybe a little bit more thought-provoking and forward-looking ideas. And that's when we came with this title towards making a molecular movie. So I'm not going to show you any rotating structures. I think the idea is really to maybe present the science that we're currently conducting in a more forward-looking way, that we can sample ourselves in a way that we create molecular movies of our circulating proteome. So when thinking about this title, and I have sort of created the theme of movie making as sort of the underlying idea. A good movie starts with a preface often, and the preface here is really what really does it take to make a movie, and what are the similarities from movie making to conducting a scientific project. So movie making often starts with an idea about a story, developing the characters, creating a synopsis with predictable turning points. Shooting the movie requires that you make a plan when to have certain scenes being recorded. Of course, you need to recruit the actors, the crew, and get financial support to be able to conduct the movie making activity. Then it's about to shoot and edit the movie, get feedback from certain audience or colleagues, and then at the end, publish and distribute. So if you think about the scientific research project and simply going along the lines that I just sort of alluded to, is of course, we need to start with a hypothesis. We have a certain biology and a set of targets we want to investigate. We hope for a specific outcome, let's say to discover a novel biomarker. And to do this, of course, we need to create a project plan. We need to collect the materials, recruit the patients, and choose a particular set of methods. And then, of course, that then leads into generating the data and analyzing the data, which then ends with interpreting the information that we have generated and hopefully submitting it to a journal. So I think there's a quite interesting similarity between very distinct and very different fields. So I think there's a lot of element we can actually learn from the two. So if you would then sort of be a little bit more self-centric, and I think we all are to some extent self-centric, is if we would make a movie of our lives, how would that sort of look, and what would be the motivations that would basically guide us in making a movie? So first and foremost, I think if we want to make a movie, this is something we often want to share, with family, friends, and maybe even the world. It's for us to preserve our memories, it's to reflect on our achievements, but maybe also to inspire others and show, these are things that are actually possible. It can also be so that we make a movie about ourselves to educate about the possibilities and maybe the decisions we have made in our lives. And for some, this can be even a sort of a therapeutic benefit. So if you think about the same thing, making a movie on a molecular level, okay, yes, it means we would be sharing our information with the doctor. We would preserve information about the molecular person that we are. It would give us information about our health trajectories. It would inspire others to maybe create new knowledge on the information. And it would help others to train, to use our data to train maybe new diagnostic or predictive models. And hopefully at the end of the day, it would teach us possibilities to intervene, let's say, with early progression of the disease. So I think there's a quite interesting similarity in these aspects, and that really sort of was the idea when I sort of created this set of slides to really show you now in the next following minutes some of the research that we are doing and some of the aims that we have that go towards these elements that I discussed with you just now. So the topics of my talk today really are, first of all, the universe. I want to talk a little bit about the circulating proteome. I want to talk about the aspects of making it personal, meaning so to profile individual phenotypes, and then to think about, how can we do things in a different way as we have been doing them for centuries, which means predominantly do clinical sampling. How can we do sampling in a more engaging way, for instance, with the general public? And then to end, to round up, to think about the next phase. So from taking singular snapshots at long intervals, maybe do this in a more sort of condensed and more narrow way that would then allow us to make these molecular movies. So as you heard, I'm part of a fantastic project called the Human Protein Atlas. And in the Human Protein Atlas project, the ambition was early on to map the protein expression across different tissues, organs, and cells. And of course, this information has then been enriched by other techniques, for instance, transcriptomics, to really better understand in which organs, tissues, or cells protein are expressed. And when you think about the subject of my talk, which is a non-cellular system, it's a system that is the circulation, meaning all the proteins that are floating in our blood, and that basically can come from all sorts of organs, tissues, cells of the body. It is a little bit of more challenging exercise because the blood that we now use for diagnostics is filled with information from different parts of our body. But as part of this protein atlas, we have also now tried to do this systematically in investigating, the sources of where proteins are coming from. For instance, the proteins that are being expressed and secreted out to the bloodstream. And today we know that out of the 2,500 proteins that are known to be secreted, about 800 are actually meant to directly go into the bloodstream, where the majority of others can, of course, still reach that sample, but through other means of transportation. So if you think about this, what we call the circulating proteome, as I mentioned, we have secretion as one of the possibilities, and we know several organs like the liver contribute more than others. But we also know that there are other ways that the protein can end up in the bloodstream. That, for instance, is the leakage, meaning when certain cells die due to apoptosis or other processes. And it can also be that certain proteins are chopped off the surface of cells that, for instance, are lining the bloodstream. So we have, some sort of categorization of these proteins, and that, of course, helps us to loop back into their functional relationship, and that helps us hopefully to better understand their role in human biology. Many of these endeavors are really heavily technology driven, and to get a sort of more global understanding of what the community is doing in terms of advancing methodologies and applying methodologies, there's something called the Human Plasma Proteome Project that, together with these fantastic people that you see here on the screen that stem from various corners of the world, we're coming together every second year to write a review about the current status of the methodologies and on the advancements that have been made in different areas with different, for instance, diseases and different technologies. And as you see, we have been doing so for the last 6 years, and now we are preparing a new version that hopefully will come to your inboxes soon. But when we think about technologies that are being used today, and here I'm specifically talking about technologies that allow us to generate hundreds or thousands of data points per individual sample, it is interesting that when we start each of these technologies with a drop of blood, we're not at the end analyzing the protein itself, but we're analyzing representative sets of information. For mass spectrometry, which is sort of the top row here, we convert a protein into peptides, and of course we ionize these peptides and then use detectors to measure these. If you think about a system that is offered by Olink, we use 2 antibodies that are modified with oligonucleotides. So we convert protein information into DNA. And similar is for the methodology that the company called SomaLogic offers, which uses aptamers as binding molecules. And again, these aptamers then convert the protein information that we have into DNA. So again, at the end, the modern technologies and those that offer the highest throughput, they actually don't directly measure proteins, they measure representatives that are much better to be sort of processed by the different analytical instruments that we have today. So when you then think about the molecular architecture of the circulating protein, I mentioned to you the different ways that this protein can end up in the circulation. I mentioned that some organs, like the liver, contribute more than others, but we can also disentangle this in a little bit of a different way. And especially when we think about the purpose of conducting analysis of the circulating protein, which is predominantly to maybe look at biomarkers. So if we think about, the leakage products, which are proteins, preferentially originating from very few organs, tissues, or cells, that could give us a very good indication about, for instance, early detection. And these proteins are usually very low abundant, very difficult to measure. So we need to be, having the right technologies that more or in a targeted way identify these out of the bloodstream. Then we have the proteins that sort of I would call the constituents of the circulating proteome, which are those that we expect to be present in blood. Preferentially, we expect those to be only present in blood. So we know that these are sets of molecules that really represent sort of the homeostasis of the blood. And this can give us a good indication about the quality of the sample that we have in hand. And this is an aspect I will come back to you shortly. And the next aspect is, of course, what does the sum of all these protein measurements actually allow us to do? It gives us an opportunity to measure health-related aspects. So we can do monitoring, we can do stratification into different groups of the individuals that we have at hand. But it's these combinations of these different sources of information that I think are important to remember, especially when we think about the purpose of using this body fluid for investigating of clinically relevant information. So unfortunately or fortunately, taking a blood sample is one thing, but the information that we generate from this blood sample is another. And that is because the sample that we take is only a proxy of the phenotype we investigate, because there are many steps in between these two. And this sort of a wheel of information, which is inspired, of course, by the Trivial Pursuit game, represents different elements that contribute to the biases that we unfortunately or fortunately start to learn about. And one bias is, of course, what is the protein that we are investigating? Because some proteins, as I mentioned, may be leaked or secreted. Some proteins may be present in all organs or just very specific ones. Some proteins may undergo, post-translational modification or interact with other proteins, which make them differentially accessible for the analytical tool that we have. The second component here is the sample type, and that really, I think, is still an underestimated contribution to the variance that we see in many of the measurements. So it really matters when a sample has been taken. For instance, if you're in a healthy or at a peak disease state, you will get a different profile just by the time when the sample has been taken. It also matters what the preparation is, because some proteins can, for instance, be found at much higher abundance in plasma preparations than in serum preparations. So knowing about how we prepared our samples are key. And of course, it matters how we store and when we have collected a sample, meaning how long the sample has been stored. Because proteins are active molecules, they tend to disintegrate over time. A third and of course very essential component is which are the individuals that we are investigating and what is their sort of contribution to these investigations. So of course we have clinical features such as age or BMI, which contribute heavily to the circulating proteome, but it's also medication that really differentiates some of the individuals just because they've been using a certain drug compared to others who didn't. Then we have learned recently, and there will be continuously more insights generated about the genetic contribution on the circulating proteome, because really it shows that the genetic makeup really has a strong impact on the levels of proteins or the interactions of proteins that we see. And of course, last but not least, it's our health history that contributes to the protein profiles that we see. So if we have lived a life without medication, exercising, eating healthy, that has had a different contribution than if we have been continuously sick, taking medications, and maybe not living a healthy lifestyle. So again, our history defines the sample that we are investigating at the given point in time. Last but not least, it's really about the design of the study. Often, samples are being collected for other purposes, and what we have been capable of doing is using these samples and sort of repurposing them for specific applications. But it's really about the design and the reproducibility of the design that needs to be considered. Because if we have a very specialized, sample and patient recruitment strategy, we need to think about how can we replicate such a recruitment strategy in a validation study, because if they do not match, we will likely generate differential information. As I mentioned, I want to spend a little time about the sample, because I think it's still an underestimated and undervalued contribution to the data that we generate. And if you simply think that a protein as an active molecule floats at 37 degrees in the circulation, and then by coincidence, a metal needle is punched through several layers of tissue, and then a representative fraction of the blood is being sucked through this needle into a plastic container at 20, that is present at 20 degrees, for example, then is being shaken and spun down and then put into smaller aliquots and then been frozen down into the freezers. So this, we hope, is a representative of the phenotype. But I think, as you can imagine, proteins as active molecules may undergo several changes just during this short period of time. But even following that collection, storage, and aliquotation, we'll have ways to distribute samples, ship them across the globe, for them because we want to use them in specific applications. And last but not least, put them into our analytical pipeline. So it really matters, what these different parameters are, and it really matters that we start to collect this information in order to understand what their contribution is. So I give you just one example here about a longitudinal study in which we investigated 100 participants through the course of 1 year. And every third month, these individuals came to the clinic and donated a blood sample. So what you can see on the left-hand side is a protein called CDL5, and on the right-hand side is a protein called caldesmone-1. On the top row, meaning here in blue and green, is the reproducibility of this assay performed on the samples at different time points. And as you can see, the reproducibility of measuring an individual sample over and over again for both these analytes is very, very high. As you can see on the left-hand side for CDL5, the longitudinal stability, meaning comparing the first with the second with the third, and the third with the fourth sample, also has an extremely high reproducibility, which means that this is a very reliable measurement and a very stable biomarker. If you look at the far more noisy plots here on the bottom right-hand side, you can see that there's basically no correlation when we investigate the sample 1 to sample 2 correlation. And that means that we cannot reliably measure this protein, even though we know that the assay is technically very good. We cannot reliably measure in resampling exercises, which means that this protein, every time we punch a needle through the tissue to collect blood sample, changes its level. And as it's been shown, this caldesmone-1 is a protein of the smooth muscle, which indicates that every time we penetrate our skin, different fractions of this protein will end up in the sample, which means that this really represents the variance that we can generate from just sampling ourselves over and over again. The next thing that we observed in this study is the same set of 100 individuals. On the left-hand side, to this sort of two-dimensional confetti plot, where each color code represents an individual, it shows that these 100 individuals, by just measuring 700 proteins, are very, very different, but they're also very stable, which means each of the individual has a specific molecular phenotype that we could measure, and we could also measure that this molecular phenotype is stable over the course of 1 year. So you remain who you are, even though some proteins may change over time. What you can see on the right-hand side is a representative group of proteins which are influenced by the genotype. So each of the color codes in these line plots indicate the different genotypes for these proteins. And you can see that in some instances, there's a very clear separation between the protein levels that we measure and the genotype, which indicates that even though the levels are very stable over time, individuals would be grouped into different categories just based on the levels that we measure. And without knowing that these different levels are due to the genotype, we may make differential assumptions about their health status. Some examples, for instance, are proteins where we have very clear differentiation between just very few individuals. So here is 2 individuals for this complement factor protein, or here we have complement factor 12. It's just 1 out of 100 individuals that has a distinctly different level of protein measured over the course of 1 year in a consistent way. So again, this really argues strongly about the fact that we need to have additional data sets to inform about the molecular measurements that we are conducting on the circulating proteome. Another interesting aspect is, of course, to follow individuals through lifestyle or health perturbations. So here are just 2 examples from these 100 individuals. Here we followed them over 6 time points, so 1.5 years. And as you can see, these sort of circular plots indicate the changes of a set of proteins throughout these different measurements. On the left-hand side, you see a more progressional change from the different time points that the person has donated samples, and it's so more of an inwards movement of these protein levels. And as a matter of fact, this is an individual that participated in the study and lost weight during the course of this trial. And because weight loss and BMI are strong factors that influence our circulating proteome, we could really measure these loss in weight and decrease in BMI just looking at these different proteins. But it's also so that if we wouldn't know about these changes on the weight scale, we probably would have misinterpreted and could have assumed other changes would occur. On the right-hand side, you can see there's just 1 out of these 6 measurements which strongly deviates from the rest. And this actually is a protein, is an indication of an infection that the person had prior to sampling. So which means that a certain event in close proximity to the sample has really sort of caused this protein, the set of proteins to change in abundance, and not knowing about this prior perturbation, we might have made the incorrect assessment of this person. So again, additional data is needed to clarify and understand the protein measurements that we conduct, but it also shows that the circulating proteome can really inform about these events, maybe in some cases, even without the person knowing. So all these examples that I've just been giving you have been conducted on samples collected in a clinical hospital. But as many of us, we have been strongly affected by the COVID pandemic, and we thought that this offers opportunities to think a little bit differently and to try these ideas with our research projects. So together with my colleague Nicholas Roxette, we came across this article early in the pandemic, which sort of brought patient-centric sampling into the context of COVID-19. And really what patient-centric sampling means is to give an individual the possibility to take the sample at home in an environment where the person doesn't have to risk, being exposed, for instance, to a virus, and sort of where we can basically create a parallel track to the clinical activities, which predominantly early in the pandemic, he really had a completely different focus than starting to sample every individual that thought that would be exposed to the virus. So patient-centric sampling is, of course, also an opportunity to test new technologies, to evolve new ideas based on the fact that can we, use this procedure to engage the general public? Can we find ways to cut costs? Can we find ways to take these small amount of samples that become available for different types of analysis? So what we've started to work with is this system by a company called Capitana here in Stockholm, Sweden. And sort of the ingenious sort of thing about this technology is that it collects a dried blood sample using this collection device, and the single drop of blood is then pulled through a capillary, which then loads a small disc at the end, and this small disc then harbors exactly 10 microliters of blood. And this precision of knowing that every sample that you investigate is loaded with exactly 10 microliters is a very big advantage, because it sort of circumvents the worries that punching out a certain blood spot from a regular Gertrude cut would create some variance. So especially if you want to look at longitudinal consistency, if you want to look at variance between individuals, knowing exactly how much material we put into our system is an important fact. So what we then sort of tested this idea on is what we call translational serology, where we measured the exposure of the virus in the general public. So early on in the pandemic, in 2020, there were no PCR testing pipelines available in Sweden, but we still wanted to go out and see how the virus has spread in the general population. So we sent out 2,000 of these blood sampling cards that I showed you on the previous slide. And we were amazed to receive 55% of these back from the general population. We were not able to give them any feedback because the assay was very, very early in the development. But of course, it allowed us to really get an unbiased survey of the population without sort of creating only measurements from those that actually are interested in the subject, that maybe have experienced that the virus has been affecting their lives. So what we found in the study published in Nature Communications is that about 12% of the population had actually been exposed to the virus during the late spring, early summer of 2020. And that when we use this multi-analyte serology based on the Luminex system, we could measure several of these informative antigens from the SARS-CoV-2 virus. So this really showed that the accuracy and precision of this tool really allowed us to get very precise measurements of the exposure. But we thought this is not the end. So we think that now we have made such collections, and at the bottom you see some of those ongoing activities that, of course, we can do home sampling. We have a way to prepare these samples, and it's, of course, a new sample type, so we need to adapt the procedures. We know that serology is really a very low-hanging fruit when it comes to investigating content of these samples. But then how can we make use of this information for stratification, and then to really apply the tools that we care most about is to measure the proteins in these dried blood spot samples. So this is what I'm sort of want to talk about during the last couple of minutes of my presentation. So really one of the thoughts here is that we have some prior knowledge about the data that we can generate. And prior knowledge is, for instance, in our case here, that we have questionnaire data where we send out with these sampling kits to the participants, and they could answer, okay, yes, I have been infected or I have been vaccinated, as this data here suggests from a follow-up study we conducted in 2021. And of course, you can use this and test your system in sort of the ways how precise and how accurate is the information that I generate. So as you can see here to the left-hand side with this representative box plots, is that the individuals that have been exposed to the virus and that have been vaccinated, of course, had a very strong immune response compared to those that have only been infected or have only been getting the vaccine. But you could also see that this is represented here by the green individuals, that some of those actually fall out of the categories that you would expect. So for instance, here you have a group of individuals that are mentioned not to be vaccinated or mentioned not to be infected, but still about 5 to 10% of those actually, represent, when we look at the molecular data, as if they would have either been, they would have been probably infected. So instead of sort of keeping these silos that, we can generate from the knowledge that exists, why don't we let the data speak to us and try to let the data inform us about the different phenotypes? And as you can see here on the right-hand side, it's the same basically underlying data, but using a different clustering methodology. We could really sort of clearly differentiate these individuals into different groups. So we had one group that had a very strong immune response. These were predominantly individuals that have been vaccinated and infected. We have some individuals that were showed immune response due to the vaccine. We had some individuals that showed immune response only due to the infection, and we showed some that had a weak or no immune response. Still within these groups, there were some individuals that, for instance, had been receiving the vaccine, but as it showed, these individuals had been receiving the vaccine quite close to the sampling date, so the immune response has not fully developed. Or it included, for instance, some individuals that had been infected very early in the pandemic, which means that there already has been a waning in the antibody levels for these individuals. So again, you get a similar picture, but still a very distinct, detailed outcome and categories that, where we want to understand human biology, might really make a difference. So of course, we wanted to take this further, and we wanted to see, well, measuring antibodies is good, and we know it's extremely reliable, and it's not only us that has shown this now over and over again. It's really to go deeper into the proteome and see, can we measure, for instance, IL-6 and TNF-alpha, which are important inflammatory proteins. And we've been using, in that case, that's about 2 years ago, the current largest set of Olink Explore assays, so here to measure about 1,500 proteins. And so we could see here, and this is just a representative illustration of the data we generate. First of all, yes, we can generate data at very high precision and high reproducibility, but also, to our surprise somewhat, we can measure nearly all of those 1,500 proteins in these dried blood spot samples. And again, these samples have not been collected in the clinic. These have been collected by a person of the general public that received a little envelope in the mailbox from us asking to contribute to our study. And then the person, after taking the sample, has just put the sampling card back into an envelope and sent it back to us in the lab. So as you can see, nearly 95% of the proteins that these different assay panels can measure were detected, which of course gives us a lot of opportunities to investigate, the different phenotypes that these proteins can represent. But it also showed that, drying proteins in the blood spot samples and then eluting them again gives you still the possibility to measure them. So of course, this is a very, very encouraging result, but this is a survey that we have conducted in a general public, which involved individuals that we cannot trace back because all of these investigations were done anonymously. So how can we take the next step? Well, we have done some research, and I can show you some unpublished data in the last 3 slides that I have here today, where we can show, the consistency of these measurements. So this is a study we've conducted together with colleagues at Karolinska Institute, and here we did a consecutive sampling over the period of almost 3 weeks, and we have dissected these into 2 different groups. So the red group would not be sort of being exposed, whereas the blue group would get an exposure. And as you can see in the beginning, for all the 3 markers, the first 2 or 3 days, there is no difference between these 2 groups, which indicates that the immune response will take some time to be measurable. But then for this feature here, you can see a very strong peak, whereas for the other feature, it takes up to 5 days until these features differentiate. And for the last one, you can see it's even less clear. So what this has sort of, provoked us to think is that, yes, if we would measure these samples for the first couple of days, there would be no difference, but still we would observe a certain degree of change, right? So that means that there is an adaptation occurring, and that this adaptation might be misinterpreted as sort of being an effect of an exposure. So again, when sort of coming back to what I talked to you about in the beginning, we need to have these consecutive measurements to learn about the commonalities between, let's say, exposure and placebo. And we need to learn about these effects that otherwise, would be misinterpreted as being a true effect. So it's really not measuring individuals or any type of samples that have been collected at larger intervals. We really need to shrink down the time between these individuals, the time of these intervals, so we can get a much more fine-grained resolution and a much better picture about the physiology. And this is, I think, really where sort of this whole movie-making idea emerged, because we need to, create more information about the transitions between these health states. For instance, if we get sampled at a regular health checkup, and then 2 years later we come back again and many things have already changed. But really, how can we do this? Because going to the clinic on a regular basis is not very convenient. So one of the ways that one can do it is to think about next generation of microsampling. And this next generation would then be minimally invasive, it would be pain-free, and it would be an easy way for everyone to sort of engage to. Because taking consecutive blood samples through the venous system is not a very pleasant way. So here's a study we did again with colleagues at KTH to look at these 1 microliter microneedle samples. And here we investigated interstitial fluid. This is again not blood, but very close to the bloodstream. And what we did here as a proof of concept was to measure antibodies against the SARS-CoV-2 virus. As we can see here to the left, this is sort of the information that we get from dried blood spot samples. So we had, representative groups that were negative or SARS-CoV-2 positive. And when we did the same measurement in the interstitial fluid, we basically repeat and confirm the different antibody levels for these individuals. So we had a clear group of negatives and positives, but you also show that here the antibody levels were about 100 times lower than in dried blood spot samples, which again puts up another burden for these delicate measurements. So what did it take to go from the snapshots, these large studies that we're doing now in using the fantastic biobanks that have been developed, to really making these molecular movies? Well, first of all, I think we will learn in the next couple of years, for instance, projects like the UK Biobank, that these large-scale analysis of the circulating proteome is possible. But we also learned that these sampling intervals, even though the studies that I showed you here every 3rd month, might still be too long, right, to really capture the molecular phenotype, and it actually might be too expensive and resource demanding. Because also, taking a venous blood sample every 3rd month is not a very convenient procedure. And you may lose in patients or individuals that are initially engaged over the course of the study. But as I hope to show you is that the timing of sampling is really crucial. So if you have individuals that come to the clinic, with high fever and high inflammation, of course, their proteome will look much different than if you measure the same individual, maybe years before or months before this visit to the clinic. I think we also have to expect heterogeneity and that fluctuations occur, because I don't think that we all are the same. And I think that will also be shown in our circulating proteomes. But really, to take this next step, the new methodologies, some of which I indicated in my last slide, should be pain-free and should be easy to use. And last but not least, when we have such material at hand, we need to have the technologies, for instance, like Olink's proximity extension assays, that really can generate a lot of information on just minimal sample volumes. And with that, there are many people that I would like to thank that you can see here on the slides, and the funding agencies that supported my research over the last decade or so. And with that, I thank you for your attention, and I'm happy to take any questions if there are. Thank you very much.